Chapter 2, Section 2, Transporting Nutrients into the Cell. Well, to get things across the membrane, well, you can get it across easier or faster, or you can get big things across if you utilize what is referred to as active transport. Active transport. Active means there's going to be work involved. So you're doing work to move things from outside the cell in or from inside the cell out. There are 3 mechanism types for active transport. There's what they refer to as a simple transport. Okay, this is just a transmembrane protein, takes things from inside, moves them out, takes things from outside, moves them in. Group translocation, where it's a series of proteins that work in conjunction with each other to move things in or move things out. And then what is referred to as an ABC system. It's 3 components. There's a binding protein, a transmembrane portion, and then a part that is all about the ATP hydrolysis. Remember, it's the hydrolysis of ATP, the removing of that 3rd phosphate, that will drive this system, hence active transport. For some of these, the simple transport group translocation, instead of using ATP, they could also be driven, the transport can be driven by proton motive force. ATP energy is used to transport out hydrogens. As the hydrogens flow back through in, they allow a transporter to move something across. So you have the sodium proton antiporter when you're looking at the simple transports. As the hydrogens flow through, it gives up whatever, is necessary power-wise to move sodium out. Lac permease, a symporter, that means as the hydrogens that were pumped out flow back in, brings along lactose. It's this flowing, and I use the term flowing for a reason, because as these hydrogens move through, they're constantly moving through, it powers this enough or allows for a change in this enough that lactose can move across. The ABC transporter, remember ABC transporter is going to have multiple portions. One of the portions is going to be the part that hydrolyzes ATP, takes ATP, turns it into A diphosphate. That is going to power the change, the conformational change of the rest of the 2 portions, a binding protein, and then the actual transmembrane portion that allows the movement across. And then a group translocator you see here just above my head. A series of proteins that will take off the inorganic phosphate off of, say, something like phosphoenolpyruvate, PEP. That inorganic phosphate that's pulled off here, it's transferred through a series of proteins that are taking that energy. And then they will finally transfer that energy to a transmembrane transporter that will then move something, say, like glucose here, across. The simple transport, as I just mentioned, can be driven by a proton motive force. So technically, they like to say that it's passive since it's not using ATP directly, but in my mind, it takes ATP to set up the proton differential, high protons outside, low protons inside, so you have something, concentration for things to flow across. The examples, as I showed, symport, antiport. The symporter, as the hydrogens flow from outside in, they allow something else to move with it. The example on the last slide was the E. coli lac permease, part of the lac operon. When E. coli gets, the signal from outside that there's a high concentration of the sugar lactose, it will trigger the expression of all the proteins in the lac operon. One of them happens to be this enzyme known as permease, which will aid in the uptake, the movement of lactose from outside the cell in. The phosphotransferase, that was the group one, okay? Taking the phosphate group, the inorganic phosphate group off of a, phosphoenol pyruvate, which is the second to the last step of glycolysis, and using that and transferring that and whatever energy is imbued within the bond with it through a series of proteins that ultimately will power a transmembrane protein that will pull across some sugar, glucose, fructose, mannose, whatever. And then that ATP binding cassette. We'll see this one pop up quite often. ABC is where they got the name. Some people seem to think ABC got that name because it's a 3-component part. No, it's the ATP binding cassette. ABC is where they got the name. One of the 3 proteins hydrolyzes ATP. Another one binds the target that needs to be pulled across. And the third player in this transport system is the transmembrane protein itself.